Bueno, buenos días. Vamos a empezar el seminario de hoy. Today we have the visit of Cyril Zipfel, who is doing a sabbatical visit to the CCG. He has been already talking to many colleagues in the department, but also people from the CCG. So Dr. Zipfel, he studied undergrad at the French University. Then he moved to do his PhD from 2001 to 2005 at the Friedrich-Münster Institute in Biomedical Research, and this was in the University of Basel. It was under the supervision of Thomas Bollert. Everybody working on plant biology knows very well to Thomas Bollert. And then he moved to the EMBO postdoctoral fellow from 2005 to 2007. And this was in the group of Jonathan Jones at the Sainsbury Laboratory in Norwich in the UK. And then from 2007 until now, he has been a group leader at the Sainsbury Laboratory, which he directed from 2014 to 2018. And since 2018, he's a professor of molecular and cellular physiology at the University of Zurich. His laboratory is mainly studying the molecular basis of plant innate immunity, and more generally, the receptors kinase-based signaling in plants, as well as how this knowledge can be used to engineer the stress-resistant crops. So he has published more than 200 papers. And he also secured, that would be nice. He also secured, ChatGPT, starting a consolidated European Research Council grant. And he's a highly cited researcher and recipient of the 2015 Charles Albert Schul Award from the American Society of Plant Biologists in 2018. Also, he got the Tsuneko Reiji Osaki Award, and he was elected to the European Molecular Biology Organization in 2018. Cyril, it's a pleasure to have you here, and thank you very much for accepting the invitation.
Thank you.
Yeah.
Okay, good. Okay, good. I'm just trying to figure out how this works. Okay, that sounds good. Good morning, everyone. Thank you for coming with this weather outside. Before I start, I would like to thank Luis, of course, for the kind introduction, and Luis and Patricia for organizing the seminar and the visit to the IBT. I really enjoy visiting the institute and discussing with people, which I did actually last week, and I'm looking forward for further discussion either today or in the next few weeks while I'm still in Mexico. So indeed, as Luis mentioned, what our group is interested in is how plants perceive the environment in a very general sense. And historically, this has been mostly in the context of immunity, so how plants basically can perceive microbes. And this was in the concept of non-self perception. So as Luis mentioned, we have been working for more than 20 years on innate immunity in plants, and mostly try to understand what are the receptors which are involved in perceiving these microbes, what are the ligands from these microbes, and how in response to this perception, then plant cells can activate defense responses that ultimately will allow them to resist pathogen. But what has been common to this research, whether it was in the context of immunity or now more recently in the context of growth, development, or even response to abiotic stresses, was our interest in a family of plant proteins, which are receptor kinases. It's one of the largest families of protein in plants. It has massively expanded in all plant genomes, and it's constitute what we call a monophyletic group. So they all have a common origin, and actually the kinase domain from these plant receptor kinases are related to kinases involved in immunity, in innate immunity, in insect, for example, in Drosophila, like Pele, or in animal like IRAC. And so to give you an idea of the numbers, in the plant model Arabidopsis thaliana, there are over 400 receptor kinases. If now you look at a crop such as rice, this number will expand to about 1,000 receptor kinases. And if you look at trees, this number could expand to several thousands of receptor kinases. So these proteins are very important, I would say, for all aspects of plant life. Whatever you're working on in the context of plant biology at some point will be related to a function of one or several receptor kinases. And the main function of these receptor kinases is that they are located at the plasma membrane of the plant cell, which is highlighted here. And they can perceive a number of signals which are either produced by the plant itself, and in this context mostly regulate growth and development, sometimes in response to environmental stresses. But in addition, they can perceive ligands which are coming directly from the environment. And this will be the case during immunity, where many of the ligands for these receptor kinases will be coming from microbes where they act as pathogen-associated molecular pattern-activated immune responses. And so the ability of this large family of plant protein to perceive a variety of different ligands is coming from a diversification of the ectodomains, which are facing the extracellular space. And this allows them to perceive basically any type of molecules. They can bind protein, they can bind peptide, lipids, sugars, or small molecules. And the largest family of plant receptor kinases contain an ectodomain, which is composed of leucine-rich repeats. And so in our group, we're interested in a number of different questions in relation to adaptation of plant to the environment. And so some of these questions are very fundamental. We want to understand how these receptor kinases can indeed perceive their ligands. So this means we want to identify ligands, we want to identify their receptors. Biochemically, we want to understand what is the molecular determinant of the binding of this ligand to this receptor kinases. Then we want to understand how, in response to this ligand perception, these receptor kinases get activated and how this activates downstream signaling. And in the last 8 to 10 years, we also started to study peptides, which are produced by the plant, which are actively secreted by the plant cell. Many of them are produced in response to stress, and how this peptide then regulate growth development in response to the environment. And as Louise mentioned, for the last 20 years, in addition to these fundamental research programs, we have also been interested to see how we can use the knowledge that we are gaining from this research foci to engineer crops which are more resistant to disease, as well as more resistant to different stresses. And I will not present this work today. So today I will just present the work here related to receptor kinase activation and how, in response to this receptor activation, plant cells activate downstream signaling. So if you think about a receptor kinase, it's a unique type of protein because within one protein, it contains different modules which have different functions. It has an extracellular domain, which is facing the extracellular space, which in plant we call the apoplast. And this is involved in ligand binding. We have then a single-pass transmembrane domain that will anchor the receptor kinase within the plasma membrane. And then inside the cell, we have an enzymatic activity, which is encoded by this kinase domain, which will need to be activated to then induce outputs in response to the inputs that will be perceived here outside the cell. So if you think it's a relatively simple question, we just want to understand how an input is translated into an output. The problem here is that these two events are separated physically in two different compartments. Here we are outside the cell, and here we are inside the cell. So not only in plants, but in other systems, this has remained a challenge to understand how this biochemical recognition here will then transduce into this chain of signaling events that will be activated within the cell. So in plant, I told you already that there is a wide diversity of receptor kinases. Some of this diversity is illustrated here. You see that we have here schematic of different receptor kinases that have different ectodomains, such as leucine-rich repeat, lectin, or malectin-like domains, and they can perceive these different type of molecules, as I mentioned before. So in our group, we work on several of these different type of receptor kinases, which are listed here. Most of the one we are working on consist of leucine-rich repeat receptor kinases, which most of them will be involved in the perception of proteins of peptides. But we also work more recently on lectin-type receptor kinases, which are involved in the perception of sugars or other type of molecules. And so for many years in the field, we wanted to understand how this ectodomain or this receptor kinases can actually biochemically perceive the ligand that they are recognizing, and biochemically what has a determinant for this perception. And so for the receptor kinases that our group has been working on, such as FLS2, which is a receptor for bacterial flagellin in the context of immunity, and it's a leucine-rich repeat receptor kinase. Or here, in the case of a receptor for a class of peptides, which are cysteine-rich peptides, which are called RALF, and regulate many different processes. And here we have a different type of receptors, which has two malectin-like domain in ectodomain. We had resorted here to structural studies using X-ray crystallography, and we had done this in collaboration with a group of Gigi Shai in Beijing. And this gave us information about how indeed, for example, in response to the binding of the ligand here that you see in pink, you form a heteromeric complex. So you have an example of ligand-induced dimerization that will then activate signaling. At least this is what is happening outside of the cell. These are just using ectodomain, which are being purified in insect cells. The problem with this kind of studies, as you might know, there are very few labs in the world that can do it, especially in the context of plant biology. It is not always easy to purify this type of protein of ectodomain so that you can obtain crystal. And now, more recently, of course, people have started to resort to cryo-EM. Still here, you need to have the right condition to express this protein. And the access to these cryo-EM facilities, especially, I would say, in the context of plant biology, is not trivial. So one thing that has changed completely the field and has rendered this kind of study, I would say, more democratic around the globe is structural prediction, such as enabled by AlphaFold. And this just happened in the last few years. And as you know, last year led to the Nobel Prize in Chemistry to some of the discoverers, at least here, of AlphaFold. And so we wanted to know if we can benefit from these structural prediction tools to understand how the ligands we are studying will be perceived by the receptor kinases. And I just show you here one example. This is another receptor that we are working on. It's a leucine-rich repeat receptor kinase. And it's perceiving a very large family of peptides which are produced by plants, which is called the scoop peptide. And it has 50 members in Arabidopsis thaliana. We try to obtain a structure, a proper structure for this ligand receptor pair, and this has not been successful using crystallography. And here we resorted to AlphaFold 3, and you can see that we could get a very nice prediction of the binding of the peptide here to the ectodomain of the receptor. You see here the binding groove. And this is published work, so I won't spend too much time, but this was among the first study where we really showed that by combining this kind of structural prediction together with phylogenomic studies, we can really predict down to the single residues, which are the residues which are determining the ligand binding, in this case of a peptide, onto a plant receptor. And for us, what was nice, this was based on structural prediction that we then validated experimentally, but a few months after we published this work, two different groups independently actually used cryo-EM to decipher the binding of the same family of peptides to the receptor, and they basically confirm the result that we obtained through this prediction. So now what we know is that here within the plasma membrane, you will have a receptor kinase, which has an ectodomain which is involved in ligand binding. And the first step in the signaling pathway is a ligand-induced dimerization with a co-receptor. And now what we know is that the co-receptor is typically short-shaped complementary, meaning that if the ligand-binding receptor contains a leucine-rich repeat ectodomain, the co-receptor will contain as well a leucine-rich repeat ectodomain, but typically it will be shorter. And so this event is critical for the activation of downstream signaling. If you block this ligand-induced dimerization, nothing will happen downstream of this event. And so then it was not surprising that our group and many groups around the world have identified that this step is very tightly controlled, but to our surprise, it is controlled by additional receptor kinases. And now different groups have identified receptor kinases within the plasma membrane that will positively regulate here this ligand-induced dimerization, and others that, in contrast, will negatively regulate this ligand-induced dimerization. And this is dynamic, so it is also reversible. And so for many years, we wanted to understand how this type of regulation was working. And this led us to doing more cell biology at the nanoscale. And what we reported now already several years ago is that this type of receptor kinases, they are not homogeneously distributed within the plasma membrane, but actually they're located into specialized nanoclusters within the plasma membrane. And within these nanoclusters, individual receptor kinase molecules are highly dynamic, and these dynamic properties within this nanocluster is important for the activity and is regulated by other components. And so this is in analogy to what has also now emerged in other fields, and for example, in animal signal transduction studies as well, where the plasma membrane is indeed not the static uniform compartment, but it's a highly dynamic compartment that forms specialized clusters at the nanoscale, which constitute, if you want, signaling platforms, which are highly specialized and regulated. So now we try to understand a bit more what will be the components here, the dynamic components of this complex formation within the plasma membrane. And at the moment in Zurich, we are doing a lot of quantitative proteomics to identify additional regulators of these particular receptor complexes. We use the immune receptors that we have been working on for many years. So I mentioned the flagellin receptor, FLS2, and here another receptor, which is a receptor for Bacteoli FTU, which is also involved in plant immunity, as model here to really understand this dynamic regulation within the plasma membrane of these receptor kinase complexes. So now, if you think about this complex here, which I told you is a minimal signaling unit, is a starting point of the signaling pathway. We have here the ligand binding receptor, the ligand that will induce the formation of a complex with a co-receptor. Now, in response to this ligand-induced dimerization, a lot of post-translation modification will happen very quickly within a few milliseconds. And the major of this event will be via phosphorylation. And then you have this dynamic phosphorylation that will lead to the activation of downstream signaling. So again, this is a step which is highly regulated. There are, for example, protein phosphatases that will regulate every kinase within the receptor complex, but also downstream in the signaling pathway. And this regulation has to be controlled. And once the signaling has been induced, there is a deactivation of the pathway by basically removing the molecules which have been activated, and this is mostly occurring through internalization, via endocytosis, and then degradation within the vacuole. So for us, in the last 10 years, what we really wanted to understand is not how this pathway was regulated somehow horizontally, so how the activity of the component were regulated, but how the pathway was acting, if you want, vertically in a classical signaling point of view. And so the first step I told you is mediated by phosphorylation. And this is why you require this ligand-induced dimerization with a co-receptor. Although the receptor has a kinase domain, by itself it is not sufficient. And this is because for this complex to be activated, you require transphosphorylation events between these different molecules of the kinase. And then in animal and the plant field, there is a lot of different models depending on which receptor you are working on or which kinase you are working on. In the context of the system we are studying, and for this type of work, we use EFR, this immune receptor, which I mentioned before, the model. It was always postulated that you must have transphosphorylation event for the pathway to be activated. And actually what we reported recently is that you have an asymmetrical activation, and that basically the co-receptor, the reason it is so strictly important or required for the activation, it is a co-receptor that basically transphosphorylates the receptor, and this transphosphorylation is required then for the activation of downstream signaling. I will not go through the details, this is all published work, but we identify specific residues that are then activated and that underlie the activation of these different receptor complex. And if you block the phosphorylation of these individual residues, you completely block the activation of all downstream events that you can measure. At the moment, we are trying to move away from this kind of work on individual domain of these receptors. A lot of this work is in vitro or based on quantitative phosphoproteomic. And at the moment, we are trying to obtain cryo-EM structure of the full receptor kinase complex. So having the ligand, the receptor kinase, full protein, and the co-receptor, so that potentially we can reveal some structural changes in response to ligand binding. So this is still something we are currently working. But now the key question that I told you before is how you activate all these molecular events that you can measure in response to this ligand binding. And now I need to bring a new component, which is here a family of kinases, which as a kinase domain are related to the kinase domain present within the receptor kinases. But in this case, these kinases are cytosolic. And so in plant, they are called receptor-like cytoplasmic kinases, or RLCKs. And so they will form a dynamic complex with the complex here of this receptor kinases. And if you wish, it is these kinases which are then activating or executing signaling in a proper sense, and then activate all these cellular events that we can measure, such as MAP kinase activation, production of reactive oxygen species, rapid ion fluxes, and so on and so forth. And so to study this, immunity is a very good system, because here, unlike plant hormones, which tend to be constantly present, and so it's very difficult to remove them so that you can study a truly dynamic process. In the context of immunity, the system is off, especially if you study in a sterile environment, which we do for some of this work. And then you can add a particular ligand, let's say a pathogen-associated molecular pattern, and you can turn on the pathway. And then once you have turned on the pathway, you can study what is happening from a few milliseconds to a second, to a few minutes, to a few hours, and so on and so forth. And we can measure many different responses in this context. We can measure phosphorylation, that will take a few milliseconds to a second, MAP kinase activation, phosphorylation of calcium-dependent protein kinases, massive transcription reprogramming within 15 to 30 minutes. Thousands of genes will be regulated in response to these solicitors, rapid ion fluxes that will occur within a few seconds to a few minutes, production of reactive oxygen species, remodeling of the cytoskeleton, and so on and so forth. And so to study here how these different events are coordinated and activated and what are the components which are involved, we are using a model system, which I've now already mentioned previously, which consists of these two leucine-rich repeat receptor kinases, which act as immune receptor for bacterial molecules. We have FLS2, which is a plant receptor for bacterial flagellin, and EFR, which is a receptor for bacterial EFTU. We know the epitopes which are recognized, so we don't need to use bacteria or purified protein. We can use peptides that mimic this epitope to activate the system under sterile condition. And then we have formation of a complex with a co-receptor. The co-receptor in this context is called BAC1. And here we have the cytosolic kinases, which I mentioned before. It's a large family of protein in plant. There are about 80 of them. But genetically, the one that is the most relevant for the pathway we are studying is called BIC1. And basically, BIC1 is known to be activated and then to activate many aspects of immune signaling in response to FLAG22 and alpha-18. And so more than 10 years ago, our group reported the first example of a substrate for BIC1 in the context of immune signaling. And we found that such substrate for BIC1 was the NADPH oxidase, RBOHD, which is involved in the production of extracellular reactive oxygen species, or ROS. And what we found is that BIC1 gets activated upstream by this receptor complex, and then BIC1 directly interact with the NADPH oxidase, transphosphorylate the NADPH oxidase, and this transphosphorylation is strictly required for the activation of this enzyme by further processes, which mostly rely, in this case, on calcium. So it's really the priming event that will basically prepare this enzyme to be later activated by other pathways. At the same time that we published this work, the group of Jiamin Zhao in Beijing reported the same findings. So this was the first example of a substrate for BIC1, and we reported more recently that this mode of regulation of an NADPH oxidase already exist in bryophyte, for example, in Mercantia, so has been conserved in land plants. So based on this work, which was performed by Yasuhiro Kadota, Yasu and then another postdoc, Shushu Jiang, observed that within the NADPH oxidase, RBOHD, the residues that were phosphorylated by BIC1 seems to lie within a particular motif. And what they observed is that you had always the serine or threonine phosphorylated in position 1 in comparison to a leucine in position 4. So they hypothesized that this constitutes a potential phosphorylation motif by BIC1, and they thought that we could search for potential BIC1 substrate by simply looking for the presence of this motif. And so this was more than 10 years ago, so we did not have access to a lot of phosphoproteomic data, but nevertheless, they screened what was available, and they identified 20 proteins that they postulated might be phosphorylated by BIC1, which then, of course, we had to experimentally validate. So this was our first attempt to predict what BIC1 substrate could be. But then in the meantime, we started a collaboration with a group working on human kinases, which is a group of Lewis Cantley, back then at Cornell Medicine in New York and now in Harvard. And he was the first group a long time ago to publish an approach which is based on the fact that kinases tend to phosphorylate linear motif within a protein. So basically, you could simplify this linear motif by synthesizing peptide, and then we can use what is called a semi-degenerate peptide array to identify in a random manner which residues would be preferred by a given kinase for interaction and phosphorylation at a particular position. And so this approach is not only was tested many years ago, but very recently was used by the same group to decipher what is the substrate specificity of the whole kinome. So we work with them to basically understand what will be the substrate specificity in vitro of BIC1. And this not only re-identifies a simple motif that we identified before, but identify additional specificity determinant within this particular peptide. And so now we could use this better motif to indeed screen within the Arabidopsis proteome and apply different filters that would increase the likelihood that these substrates are involved in immunity to identify 77 candidate BIC1 substrate protein that we call CBSPs. And so now for the rest of my talk, I will just present you some example of this BIC1 substrate and to show you what we have learned through this approach, which started from a very biochemical point of view. I will show you how some of these substrates regulate the activation of the complex, how others regulate ion signaling, while others identify completely uncharacterized kinases in plant and their important role in the context of immunity, and uncover a new mode of regulation between the two layers of plant immunity, which I will explain better later. So now, if I come back here to the receptor complex at the plasma membrane, I told you already before, but in response to this ligand-induced dimerization with a co-receptor, there will be phosphorylation event here within the receptor complex, and this is required for activate downstream signaling. And so as in many cases in plant signaling, this step is under negative regulation. And 10 years ago, we identified a class of protein phosphatases, which are PP2Cs, which are called PLL4 and 5, which are negative regulator of this receptor kinase, receptor complex activation. And at least we showed that before genetically. But now we identify that the same protein phosphatases are actually direct substrate of BIC1. So what this work showed is that these phosphatases are here to block the hyperactivation of the receptor complex, and upon ligand perception and activation of the receptor, BIC1 transphosphorylate the phosphatase, and what we found is that in response of this phosphorylation, the phosphatase dissociate from the receptor complex, enabling the full activation of downstream signaling. So this is what we report in the context of immune signaling. But while we're doing this work, we discovered that two other groups, one group in Switzerland, Christian Hartke, and one group in US, Zach Nimschuk, had identified paralogs of the same phosphatases in another context, in a context of development, which is mediated by leucine-rich repeat receptor kinases and some peptide, which I call CLE. And here the mode of regulation was basically the same. So what this work illustrated is that across different receptor kinases, which play different function, either in the context of immunity or in the context of development, and in response to different type of ligand, in this case here, ligands coming from microbes, and here ligand coming from plant, the mode of regulation of the activation of this complex is actually conserved. But now for us, what was the most interesting was to try to understand how at the molecular level all these events that we can measure in response to elicitor perception are actually occurring, so what are the components involved, and how are they regulated. And so in the context of this work, we identify a transporter which is directly phosphorylated by BIC1, which is a phosphate transporter. And this is something we did not expect initially. We expected to identify channels involving calcium influx, or here we had shown before that BIC1 phosphorylated via ADP oxidase. But here we identify this major phosphate transporter, which is called PHT1, and we identify two isoforms as being phosphorylated by BIC1. And so we wanted to study mechanistically what was the relevance, the biological relevance of this phosphorylation. And we developed an assay in root hair, in bulging root hair of Arabidopsis seedling, to measure in real time this transport of inorganic phosphate. And what this allows us to do is to measure what was the impact of elicitor treatment. So if we add to the plant FLAG22 and alpha-18, and to our surprise, we found that in response to elicitor treatment, phosphate transport was very rapidly inhibited, like within a few seconds. And this inhibition was dependent on BIC1 and the phosphorylation by BIC1. So this means that the rapid inhibition of phosphate transport is one of the earliest responses that we can measure in response to elicitor treatment. And at the time, we didn't really know how to make sense of that. But different groups started to publish work on phosphate transport in different contexts of plant-microbe interaction. And for example, there were reports that phosphate transport was an important component of shaping the proper microbiome of Arabidopsis. And in collaboration with Gabriel Castrillo, we could indeed show that this mode of regulation by BIC1 was important for the proper shaping of the Arabidopsis microbiome. And then in collaboration with Alberto Macho, we look at the role of this inhibition of phosphate transport in the response against a root-infecting bacterium, which is called Ralstonia solanacearum. And indeed, this mode of regulation is critical to mount resistance against Ralstonia. So this unexpected response turned out indeed to be relevant in the context of interaction of plant with different microbes. So for us, one response which has always been something we wanted to address was this rapid influx of calcium. And to give you an idea, this is a response that many labs have been using for more than 20 years as a readout to measure elicitor activity, to identify novel elicitors, to identify receptors. But the question was always, what is or what are the calcium involved in this rapid influx? Many people have tried genetic screen. It has not led to anything. What we know is that genetically, this rapid influx of calcium depends on BIC1. It was published by our group as well as different groups as illustrated here, and is not specific to the elicitor you are using. This is FLAG22, this is alpha-18, this is a stress-regulated peptide, and this is fungal chitin. And so we were curious to know if within the predicted BIC1 substrate, there could be protein that could act as calcium channels. And then we started to work on a protein, which was a very nice substrate biochemically. And while we were working on this protein, it was discovered that this protein was part of a newly identified conserved class of calcium channels, which is conserved across eukaryotes, which are called the OSCA. And what we found is that one member of this family, which is called OSCA 1.3, is indeed a direct substrate of BIC1. And this phosphorylation of OSCA 1.3 by BIC1 is required for the activation of the calcium channel activity mediated by OSCA 1.3. So this was all nice, but the problem is that the role of OSCA 1.3 in this regulation was specific to one cell type within the plant. It was specific to guard cell. And what we found is that this mode of regulation is actually required for the response of the guard cell in response to elicitors, where in response to elicitor treatment, the two guard cells that form the stomate are closing to potentially restrict the entry of the pathogen. So this was nice, but Oscar 1.3 was not required for the response in terms of calcium influx in response to elicitor in other parts of the leaves, like the mesophyll or the epidermis. And you can see here, genetically, we can knock out these two channels. There is no difference compared to wild type. So this told us that there must be other channels involved in these other cells involved in this rapid calcium influx. And so in 2019, the group of Shen Luan in UC Berkeley in US published two different calcium channels which belong to a different family of calcium channels, which are called the CNGCs, and reported that CNGC2 and 4 are substrate of BIC1 and are regulated by BIC1 and participate in this early response, which is this rapid influx of calcium in response to elicitors such as FLAG22. This is now cited in all reviews as being the channels being involved downstream of FLS2 involved in this rapid calcium influx. The problem, if you read this paper carefully, the phenotype they report is only present in very specific conditions of specific concentration of external calcium. If in normal condition, you go to the plant and you look for the dependency on BIC1, as I showed you before, BIC1 is genetically required for this response, but mutant in 2 and 4, the CMG2 and 4, are not affected. And this could be explained by previous studies, which were not cited in this work, showing that this mutant have indeed pleiotropic phenotypes, which are most likely due to the function of these two channels in the homeostasis, if you want, of calcium, and therefore it might not be related to a function in immunity. So while we did the screen, we, of course, look at the CNGCs because it was a proposed family of potential calcium channels involved in immunity. And these are 2 and 4, which I mentioned before, that were published by the group of Sheng Luan. And we found 2 other members of the family, 19 and 20, that contain this BIC1 phosphorylation motif. And you see that 2 and 4 do not contain this motif. So biochemically, they cannot be phosphorylated by BIC1. We also test that experimentally, and we found in a number of different assays that CNGC 19 and 20 are indeed proper substrate of BIC1, while CNGC 2 and 4 are not. They cannot be phosphorylated by BIC1. We then generated mutant for CNGC 19 and 20, and unfortunately, we didn't see any phenotype. They were not affected in the rapid calcium influx. They were not affected in ROS production. They were not affected in plasma membrane vaporization. And we're not only the only one finding this result. 2 other groups, the group of Jiamin Zhao in China and the group of Li Boshan and Ping He in US, as well tested this double mutant, independent double mutant, and they could not observe any phenotype. So this was very frustrating for us. We spent many years trying to document a potential phenotype. But then we were studying something else in the lab. I told you that we became interested in these peptides, which are produced in response to stress. And while we're characterizing some of these peptides, such as the scoops, which I mentioned before, we observed that all elicitors that we are working on, they induce a biphasic ROS production. And when I say biphasic here, I'm not talking 1 and 2 minutes. What I'm saying is that you have a ROS burst that will occur within a few minutes and typically peak in after 20 minutes. And then at 3 hours after treatment, you have a second late ROS production. It's always at 3 hours that it is peaking. So all the elicitors which are listed here, they can induce this biphasic ROS production. But in response to scoop peptide, you see that the amplitude is massive in comparison to the other elicitors. And this does not correlate to a higher initial ROS burst. And so we wanted to understand a bit more the component involved in this biphasic ROS response and also in the potential higher amplitude here in the one mediated by scoops. And just to give you some background, this is our work on scoops. So I told you this is a family of peptides which are produced in response to stress. And I already showed you before that we identify the receptor. It's a leucine-rich repeat receptor called MYC2, and this is a work which I cited before. So what we found is that this second late ROS production, this is the reason I mentioned it here, surprisingly is dependent on CNGC 19 and 20, and I'm not showing you the result here, is dependent on the BIC1-mediated phosphorylation of CNGC 19 and 20. It's not unique to one particular scoop. All the scoops we tested have this genetic dependency on CNGC 19 and 20, and the other elicitors which as well induce the second peak, you see that the second peak is, however, not dependent on these two calcium channels. So I wish I could tell you more about why this is the case, but we still don't know. And it's still something we are working on. And at the moment, what we're interested to look at is that in Arabidopsis and in other plants, such as legumes, like in lotus, for example, CNGC 1920 or the orthologs have been linked to phenotypes that are related to cell death. And so we wonder if there is not a link between this late huge ROS production induced by scoop and the ability to induce cell death, and some of the components which are involved in the execution of the cell death, potentially the CNGC 1920, but as well potentially other receptor kinases are involved, and this is something we are currently working on. But in summary, what we have found in the context here of calcium is that this subgroup of CNGCs, 2, 4, 19, and 20, they are not involved in early immune signaling. What we found, however, is that two CNGCs from this subgroup 19 and 20 seem to be involved in this late response induced by certain peptide, which are stress regulated, and we wonder if this is not linked to the activation of cell death, and this is something we are studying. But the problem, the question remains the same. If the OSCA and this particular CNGC do not participate in this very rapid calcium burst, then what are the channels which are doing this particular job? And so there are many candidates in the literature. Of course, we are not giving up on the CNGCs, and it's still possible, but other CNGCs are involved. There have been reports of anchoring repeat proteins, such as ACD6 in Arabidopsis or LR14A in wheat, that could fulfill such roles. There are reports about annexin as a strange class of calcium channels that could be involved. Another class of immune receptors, which are NLRs, nucleotide binding leucine-rich repeat proteins, have been shown to form calcium permeable channels, so they could be involved. Or, as in the case of OSCA, maybe there are new uncharacterized family of calcium channels in plants that still remain to be discovered. And of course, it is possible that it's not or, but actually all together they contribute to this response. And so this is something we're actively working on. So now I just want to briefly show you other substrates that we identify for this approach. And just to show you that without this approach, which was unbiased, looking at the presence of this motif, we actually identify proteins that otherwise would never have identified as being involved in immunity. When we look at the list of the 77 candidate substrate proteins, 8 of them belong to a family of proteins which is uncharacterized in plants, and it's a family of kinases. These kinases are called cycling-dependent kinase-like protein, or CDKs. And it's really confusing because most of you will be, of course, they are characterized, they're involved in cell cycle. No, they are called likes, and biochemically, they're actually not related to cycling-dependent kinases, CDKs, and this is illustrated here. They represent a different branch of these kinases, which are also not related to yeast or human CDKs, and they do not bind cycling. What we found is that many members of this family contain here a predicted BIC1 phosphorylation motif, which is highlighted in blue, and for all of them, we have confirmed that they are excellent substrate of BIC1 in vitro, and we have confirmed in vivo as well the phosphorylation by BIC1 in response to elicitors. Then we generated mutant for some members of this family, and what we found is that when we generate knockout of two members, such as CDKL5 and CDKL6, we see hyper-responsiveness to different elicitors, and this correlates to an increase in disease resistance against a number of different pathogens. So genetically, at the moment, what we can say is that the CDKs are a negative regulator of plant immunity, and now, because it's a mostly uncharacterized family of plant kinases, we of course try to understand at the molecular level how do they work. We are trying to identify proteins that form complexes with them, and also to identify what could be the substrate. Now we were left with about 50 proteins that were candidate BIC1 substrate. And of course, what we could do, we can say, okay, one PhD student work on this one postdoc work on this one, and for the next 15 years, that's what we do. But we decided to basically step back and to use somehow a systems approach. What we did is that, or Ryan did, was a very ambitious PhD student. He's still a PhD student. He decided to basically CRISPR all the remaining candidates and to generate not only single mutant, but multiple mutants for closely related protein, looking at the phylogeny for every genes that encode this particular substrate. At the same time, he confirmed biochemically that all this protein can indeed be phosphorylated by BIC1 in a site-specific manner. And then he performed a genetic screen, basically, using this population of CRISPR mutant, where he looked for altered susceptibility or resistance to two different types of pathogen, a bacterium, which is a hemibiotroph, Pseudomonas, and a fungus, Botrytis cinerea, as a necrotroph. And then he used a representation here to illustrate the data, where you see here the susceptibility of a given mutant in accordance to the score as a biochemical substrate of BIC1. So basically, a mutant which is located here is hypersusceptible, in this case, to Pseudomonas, and at the same time is a very good substrate of BIC1. So for us, this would be a perfect then protein to continue to characterize. And so now we are basically going through these different members, and we have identified our component of immunity using this process. I just want to finish briefly, because this is a very hot topic in plant biology, about the interplay between these two layers of immune perception. We have been working historically on this recognition here at the plasma membrane, which I explained you before. Many other groups are working on this intracellular immune perception, which involve these immune receptors, which are called NLRs, which I mentioned before, these nucleotide binding leucine-rich repeat receptors, which are related to NOD proteins in mammals. And so it has been published by different groups in the last 2 or 3 years that these two layers of immunity do not work in isolation, but actually there is a lot of crosstalk between these two layers, and there is a mutual potentiation between these two layers. But this phenomenon has been reported, but the actual component involved in this crosstalk are still unknown. And so we wondered whether BIC1 could play a role in this particular crosstalk. And the reason we wondered about this is because within the protein that we predicted to be BIC1 substrate, we found some of these intracellular immune receptors. We validated biochemically that indeed they can associate with BIC1, and we also validated that they can be phosphorylated by BIC1. And this was a work from a postdoc, Sarah Choi, who has since left the lab. So then we wanted to know what is the relevance of this phosphorylation, and this is our hypothesis, was that in response to BIC1 phosphorylation, you would activate the NLRs, because first microbes would be perceived at the plasma membrane, so it made some more sense that this helped then these intracellular immune receptors to be more active. But we found completely the opposite. What we found is if you mimic the phosphorylation of these particular residues which are targeted by BIC1, you actually block the function of these NLRs. And one of the outputs you can measure is cell death. So when you see here a dark patch, this is representing the death of the cells within this patch. And if we infiltrate here Nicotiana bantamiana, I forgot to say, with a variant of this NLR where we made a phosphomimetic mutation for this particular serine, we block the activation of cell death. And this is for all the NLRs that we have tested for, that can be phosphorylated by BIC1. And this is also happening in our Arabidopsis. So now biochemically, we are mostly biochemists. We wanted to understand how this inhibition was actually operating. And it has been reported in recent years that NLRs, the activation requires oligomerization. And you can visualize this oligomerization using blue native page gels. And you can see that in response to the activation of this particular NLRs, you see the formation of oligomeric complexes. But if you mutate one residue and you mimic the phosphorylation of this residue, you completely abolish the oligomerization of this particular NLR. So it is the first step of the activation, which is mediated by this oligomerization, which is regulated by BIC1 phosphorylation. So now you might ask, if this negative regulation occurs, how do you get rid of it to activate immunity? because these two layers do get activated at the same time. And what we found is that in response to elicitor perception, PIC1 basically dissociates from these NLRs. So our current model is that in a naive state, in the absence of microbes, PIC1 constitutively form a complex with the NLR, transphosphorylate the NLRs which block the oligomerization. Now in response to elicitor perception at the plasma membrane, PIC1 get activated and then basically get dissociated from the NLRs which enable the oligomerization. There's still a lot of question linked to this model. We need to see how general it is. Now I just show you with a few examples with a couple of NLRs. What are this actual mechanism of this oligomerization regulation? And more importantly, it's one thing that we remove PIC1 from the complex, but you need to remove the phosphorylation. And so we postulate that there must be specialized protein phosphatases, which are involved here, that are activated in response to immunity to dephosphorylate the NLRs so they can get fully activated. So the work I showed you here was really work from many people in the lab, which I acknowledge in the slide, which took us over 10 years. And I showed you there are certain things that we had expected, and basically this work confirmed the role of the importance of some of these components. We identified novel components, a novel mode of regulation that somehow were expected, like phosphorylation of NADPH oxidase, the fact that some calcium channel would be directly regulated by PIC1, and so on and so forth. But we also identify components of regulation that we would never identify without this approach, such as the inhibition of phosphate influx as a rapid immune response, the identification of CDKLs as negative regulator of immunity, the inhibition of NLR activation, and we have many other things that this screen has allowed us to discover. So, of course, I want to thank all the people who did this work. They are listed here over the last 10 years. We had many collaborators involved in this work. And there's one person I need to thank in particular, is Tom DeFalco, who really led this whole PIC1 substrate screen in the lab, and is now assistant professor at the University of Western in Canada. And this work was mostly funded by the European Union, by the European Research Council. And I thank you for your attention.
Thank you, Cyril. Questions?
Hi, Cyril. Very nice talk. I just want to know what type of NLRs did you test?
So it's not specific to one type. So for the non-immunity specialists, there are 3 types of NLRs in plant, the CNL, the TNL, and the RNL. And we found phosphorylation of all 3 types. So it's not specific to one class. Yeah.
And I guess you have already tried mutants.
So the mutant that we have, the one I showed you here, are either in TNL or CNLs. But it's not shown here. We also tested helper NLR, so RNL, and we see the same mode of regulation. So in all the cases, the phosphorylation by PIC1 is actually inhibiting the NLR activation, which is not what we had expected.
No, that's very, very nice result. Okay, thank you.
Hi, Cyril. Impressive. I have a question about the co-receptors. Is there always the same co-receptor with one? Or you can have multiple co-receptors, and then whether that will change the specificity of the kinase downstream and the output?
Yeah. So there are multiple levels of the answer. Is it always the same co-receptor? No. The co-receptor type depends on the type of the receptor. So a leucine-rich repeat receptor kinase would only form a complex with a leucine-rich repeat co-receptor. Now, if you consider leucine-rich repeat receptor kinase, the majority of co-receptors which have been reported belong to a family which are called the SERCs. In Arabidopsis thaliana, there are 5 members. And if you consider a single complex, one molecule of the receptor will only form a complex with one circ. Genetically, in the pathway we are studying, one of these circs, which is called circ3 or BAC1, is the most relevant. But we have shown many years ago that the other circs can also form a complex, and so there is functional redundancy. Now, for some receptor kinases, they seem to have a preference for one circ or another. FLS2 will prefer BAC1. EFR doesn't care so much. It can do BAC1, it can do circ1. HISA, which is another type of receptor, prefers circ1, for example. And so this might contribute to signaling specificity, as you said.
Here, thank you for your nice talk. I have 3 questions. One is, it is known that Feronia and Ralph needs to form condensates with extracellular, it's a extracellular condensate with pectins. Mm-hmm. You know whether some of these receptors or complexes are forming condensates extra or intracellular? Yeah,
so I mean, for Feronia, this is mostly the work from Alice Chung, where she showed that in response to Ralph perception, we have the formation of these clusters at the membrane and then internalization. So all receptor kinases, this is my point of view, in response to binding their own ligand, will get internalized. Now there's a lot of discussion, is it part of signaling? Some people think. What I think, it's basically just part of getting rid of what has been used. And so, in context to your question, all receptor kinases, upon ligand binding, you will observe some condensate because this is the state prior to internalization. I see. So it's just a focal concentration, I see. If you want. Yeah.
Okay. The other question is, you mentioned that you're interested in doing cryo-EM for these complexes. But my question is whether you're including membranes in this analysis.
So we are using 2 approaches at the moment. It's very challenging, so I don't have a lot of data to show you. We are using human cells, so HEK cells, human embryonic kidney cells, and we are using also a plant-based system, which is Nicotiana bentamiana. We have primary success with both, and so in this case, we express the full receptor kinases together with the core receptor and the ligand, and we extract the native membrane environment. Of course, in the context of HEK cells, the membrane is different than the plant context, so we would prefer to use a plant-based system, and so now we are prioritizing Nicotiana bentamiana for this.
Oh, yeah, okay. Yes. And the last one has to do, how do you vision the activation of the phosphatases?
In the last part I showed you?
Part, yeah.
Yeah. So here, I mean, this is just a model at the moment. So I would not be surprised if the phosphatase is actually as well phosphorylated by BIC1. And so now we are looking at, within our substrate, if there is any phosphatase, known or unknown one that we might have missed. And from the literature, we have candidates as well. There have been phosphatases which have not necessarily been linked to this first step of immunity, but actually this later one. And so now we are testing if it could play a role here in the oligomerization and the activation of the NLRs.
Okay. Thank you.
If we don't have any further question, please join me to thank
Thank you very much.
Thank you, Cyril.